Okay, we can get started. Welcome to the first CHRC webinar in 2022. I'm co-director Li Tian. We have two industry colleagues presenting today, Chris Polly and Caleb King, both from Front Range Biosciences. So Professor Dong Land in the chemistry department will be introducing the speakers. Dan, go ahead. You're on mute, Dan.
I was just talking. I don't know how that got muted. Okay, so Chris and Caleb, I've known now for many years and worked with in a lot of different ways, both at Steep Hill, which was a cannabis testing and R&D company that started in about 2008 and is still in doing testing all over. The R&D portion of that was where Caleb and Christopher and I interacted the most, although Caleb started out in the Steep Hill, Hawaii licensee version. So he moved from Hawaii to California and then started working for R&D at Steep Hill Corporate. And then the research and development team is made up of a dozen or so scientists in chemistry, genetics, bioinformatics, and chemometrics, and agricultural agronomics. So all different phases, including field trials and so on. So excuse me, Chris is got his bachelor's degree from Colorado Boulder, working with genetics and bioinformatics there, and Caleb has a bachelor's in chemistry and a master's in geochemistry, or did I flip those? I think I got it right. At any rate, they're going to talk about the use of both chemical testing and genetic testing and the bioinformatics to understand the traits, propagation, and hybridization models. All right. Take it away, guys.
Thanks, Don, for the introduction. As Don said, I'm Caleb King. I'll be doing the first half of this talk, primarily based on metabolomics as a method of breeding for cannabis. And I'm going to refer to both hemp and adult use when I say cannabis in this talk. Chris Polly will take over the second portion of the presentation, where he'll be bringing to you discussions about how genetic tests can be utilized in tandem with some of the chemistry to breed new cultivars of cannabis. I'm able to share a lot of these insights to you today about metabolomics, largely due to the incredible research and development team that Don mentioned here at Front Range. We have many members on our team with tremendous contributions over the last decades in cannabis science, and those expertises range from agronomy to pharmacognosy. By the end of this presentation, I'm hopeful that everyone will be familiarized with how cannabis can avoid some of the pitfalls that other agricultural commodities have experienced in the last century as a result of uninformed breeding decisions. And some of those breeding decisions largely based upon just how much product can actually be made, how much of an agricultural commodity can actually be produced. Try the next slide here.
You just have to hit next slide.
Okay. I think it's important before we begin to dive into metabolomics, really to understand what it is and how other omics-based approaches can really help us identify biochemical pathways in organisms, such as in this talk, cannabis. And so illustrated on this slide, we have the metabolomics and how it's combined with other omics, such as proteomics, genomics, and transcriptomics to understand the phenotype of a plant. And more specifically for cannabis, I'm going to refer to a chemotype of a plant. And so by relating some of these changes in chemotypes under optimal environmental conditions, and then under stressful environmental conditions, such as the biotic and the abiotic stressors shown here, we can make informed decisions on how to improve a lineage, a genetic lineage. And so metabolomics is the study of what molecules have been produced by this phenotype, giving us some historical context of the plant environment and what its environmental stimuli that it responded to. Once you understand the metabolome, you can then go and understand the other omics and how they can play into this. So for instance, and Chris will talk about this later in the talk, is how genomics can be used to help us predict what will be observed in a phenotype. What are we expecting to get out of our crosses? Then you have transcriptomics and proteomics, which tell us more about the present moment for the plant, or what is being transcribed from the DNA to give us a phenotype or a chemotype. So if changes occur in a phenotype as a result of environmental stressors, it's often an indicator that a metabolic pathway has also changed, largely due to in the form of its DNA or its RNA and even proteins. And so all of that combined, hence that's how the whole phenotypic response to abiotic-biotic pressures then goes back around to the systems biology paradigm, and you approach it all over again. So before we begin diving into cannabis breeding with metabolomics, might be helpful to provide evidence where metabolomics has been employed for other breeding purposes. And one of those agricultural commodities that it's been used for in the past is tomatoes. And I love the story of tomatoes because it's undergone significant breeding efforts in the last century to yield popular store varieties such as Roma tomatoes, cherry tomatoes, and the heirloom variety shown here. And that was largely thanks to, as I'm going to go into, the metabolomics, understanding the metabolome of the tomato plant, as well as the genetics of the tomato plant and how the genetics can help you make informed decisions faster. This graph shows the tomato as the example here, with genetic erosion occurring, or that loss of genetic diversity through domestication and intensive breeding. And this occurred pre-1960. And the most common phenotypic trait before the 1960s that people tried to really get was crop yield. That was cared about the most. Notice how genetic diversity in the tomato populations decreased. And so at that decreasing point, it also happened to coincide with the resistance, a decreased resistance to biotic stressors, such as insects and pathogens that were going around the United States at the time. Our solution to that as a country was to put down widespread use of insecticides, herbicides, and fungicides, all to support our growing population at the time. Other agricultural commodities experienced similar issues as the tomato plant in the last century, and some of those crops include maize, wheat, peas, and barley. Fortunately, around the 1970s, 1980s, we had this genetic diversity increase in tomato populations and in other crops, as you'll see. And what this was largely based upon was the research efforts towards agricultural breeding that included the chemistry, the color, the smell, and the resistance of these plants to biotic and abiotic stressors. And this was in addition to looking at the yield of these tomato plants. So the reintroduction of the genetic diversity back into the population required the acquisition of heirloom varieties and other landrace germplasms. Translating some of this genetic diversity index now over to some of the chemistry on the plant, we see similar trends occurring. And so to start here at a basic level, this graph shows that how the size and the dimensions and the water content of tomatoes increased from the 1960s to the late 2000s. The larger circles correspond to juicier, bigger tomatoes with overall dimensions. So, this introduction also, it's important to see that as you got not only bigger tomatoes and juicier tomatoes as time went on, there's also a small little line of dots that you'll see in this graph at the bottom in between 1990 and 2010. And those represent the introduction of the cherry tomato varieties into breeding germplasms at the time. The second figure here investigates more the primary metabolite evolution of tomato plants. And so primary metabolites being citric acid cycle, shikime pathway for amino acids, base level things that are needed for the plant to survive. And so the graph here shows the sugar to acid ratio. And again, the introduction of the cherry tomato around the 1980s is evident in this figure, because you see the total sugar content of varieties start to tick up while the acidity and the bitter peanut polyphenols went down. The third figure then takes the genetic diversity discussion here one step further, and we examine the secondary metabolites. And specifically secondary metabolites that are volatile organic carbons, those that really have a distinct smell. They aren't considered essential for plant function, but they definitely have a role in giving the taste and the smell that we're used to in our stores, as well as some of the crop resistance to water and drought and other biotic stressors that this plant occurs. So these metabolites served as biomarkers, these volatile organic carbons served as biomarkers for breeding tomato varieties that were more desired and more preferred by customers. It's easy to see from these graphs that tomato breeding resulted in not only phenotypic differences, so bigger tomato plants and bigger yields, but it also had changes to the primary and secondary metabolites as well. I'm going to transition here to cannabis, where the verdict on total genetic diversity is still out. We have some clues. At least here in the United States, we have an idea of what might be going on with the population of cannabis. Specifically, this graph here is the average THC content of a cannabis flower confiscated by law enforcement in between 1995 and 2014. And very clearly, you can see that the potency of cannabis has gone up over time. And this increasing of the THC content, which is to say you have more potent flowers or more potent varieties being released out there, it really begs the question, what kind of effect are we having on cannabis as a population? Or what effects is the agricultural commodity of cannabis having for its long term? What are we going to experience? And in seeing the THC go up, you have to wonder, what else are you also losing as part of this inbreeding that we're doing to breed higher THC plants? And so that's what I'm going to touch on, is what are these other compound classes and why are they important to study? And so we'll go through more specifically the secondary metabolites of cannabis, not so much the primary metabolites. And so we'll go from top to bottom, or the above ground biomass to the below ground biomass, with this first chart delineating 175 different compounds in a CBD cultivar. And this was for a flower only. And you can see that this graph here shows that the majority of the compounds in a flower are terpenoids and cannabinoids. They don't make up everything. And so we also have other compounds, other lipid compounds, alkaloids, flavonoids, that make up the resinous part of a cannabis plant. It's important to note that this graph could look vastly different for a different cultivar. So this is just for one CBD lineage. We move down the plant now into the stalks, and in particular, when I reference the stalks, I'm referring to industrial hemp here in this discussion. The graph on the far right shows how that it is primarily composed of hemicellulose and cellulose. And these don't account for 100% of the stem's total dry weight. They account for a majority of it, but not all of it. And so there's other compounds that are important in the stem for its rigidity or its resistance to UV damage, growth pressures, any number of factors that I can go into. These secondary metabolites, such as sterols and triterpenoids, play a role in supporting the cannabis plant. Moving even further below the plant, now down to the below ground biomass, we have secondary metabolites in the roots. Roots are mostly lignin and cellulose, but not all. And so the triterpenoids compose a large percentage of the secondary metabolites of the roots, specifically one of the compounds being fridolin. If you ever have the opportunity, I encourage you to harvest a hemp plant immediately, smell the roots, and understand that smell and see that there's definitely more to this plant than just the above ground biomass. We already know that this below ground production of secondary metabolites in other organisms such as Arabidopsis is really crucial for what's going to happen in the above ground biomass. So we have a whole other aspect to this cannabis plant to look into beyond just the therapeutic reasons, but we also need to look into the below ground biomass for breeding purposes as well. So I've talked about what we can get out of this plant. What are all the different, what's the metabolome look like? How do you now measure those biosynthetic products and some of those intermediates? And even beyond that, how do you calculate fluxes of those compounds, the movement in and out of them into a cell? And that's where instrumentation such as GCMS, LCMS, FTIR, and NMR are traditionally used. Each of these instruments have a specialized atomic range, which they can detect molecules of different sizes, different functional groups, and they all have their own important purpose for analytical testing for cannabis. So in particular, if we look at a GCMS, it primarily tests the smaller molecules, those that are volatile compounds, hydrocarbons, including esters, ketones, alcohols, and even amino acids. The next kind of phase up from a GCMS is your LCMS, or a liquid chromatography system coupled to a mass spectrometer. And that's capable of quantifying medium-sized molecules to larger-sized molecules, including vitamins, polyphenols, lipids, aka cannabinoids. And so on a identification level and a quantification level, these are excellent and really common instruments used in metabolomics. In terms of obtaining a large chemical data set quickly and even less of a cost than GC and LC methods, FTIR is a really powerful tool in this, and this can be for metabolomics. And that's illustrated by the graph here, where there's spectrums that are shown for plants at different phosphorus concentrations, or I'm sorry, different potassium concentrations. And so how does that spectra change? What are the volatile compounds? What are the compounds in general that are produced in their functional groups? The limitation, though, to FTIR is that it has limited molecular specificity. And then lastly, you have NMR, which is an excellent instrument for structural elucidation of unknown compounds. And it's even critical in helping isotopic labeling studies. So to determine how much of a compound is flowing in and out of a cell, that's where NMR can really come in handy. So the key to successful metabolomics is obviously collecting the right analytical data and also collecting enough of it to make an appropriate conclusion. And it often tends that you have to collect a lot of analytical data to make correlations and anti-correlations that are meaningful. And so if the analytical data is valid, you can generate for cannabis. This is one of our team members produced was the correlations and anti-correlations for terpenes in cannabis. And so these basic statistical techniques, these one-dimensional or dimensional plots of correlations, where the blue circles here indicate positive correlations and the red circles indicate negative or anti-correlations. An example here for cannabis, a terpene in cannabis, would be of an anti-correlation would be valencyne. Valencyne is not typically found with alpha and beta farnesene. And so with plants, why would that be? Understanding why those two are not typically found together initially raise for good hypothesis questioning. An example of a correlation then would be humulene and caryophyllene. And the correlation's excellent between the two. And again, begs the question, why might that be? And if you go and can do some functional characterization, some clonal work, you can explain this correlation through biosynthesis. And the biosynthesis, Chris is going to talk on the cloning and functional characterization aspect, but the biosynthesis now tells you a molecular production of these compounds. How are they produced? And in what ratio then are they produced? From there too, you have the enzyme that's responsible for producing the products. How can you tailor that enzyme to produce one or more of the other? So ultimately, understanding and breeding for some of these cannabis cultivars really requires a good understanding of the enzymes and the gene families that are responsible for making those enzymes. And so we can reverse engineer some of these metabolic pathways even further. Again, taking lots of chemical data here, that is the key thing I have to emphasize, is using lots of chemical data and advanced statistical techniques such as hierarchical clustering in the top graph and principal component analysis in the bottom graph. And the hierarchical clustering being really able to tell you lineage-specific metabolic profiles, or what lineages tend to have more of a certain terpene versus another lineage. And in that, you can identify certain lineages that are rare, those that produce a set of all of the compounds. And that's just by looking where, in that top graph, for example, looking where the blue squares are predominant and in what varieties they're predominant in. Jumping down to PCA, it makes it really possible to extract a lot of analytical information and group samples and make similarities and differences between them. And so in grouping them, you can create clade structures, you can create chemotypes. And so it's being able to categorize cannabis into lineages that then are important for breeding decisions, because plants that have certain terpenes are ultimately able to withstand certain environmental conditions better than other plants. And that same is true not only for terpenes, but for flavonoids and these other compounds that cannabis produces. So it's able to tease out, along with experimental studies, what is environmentally caused versus what is the true genetic cause of this plant producing a compound. The overall goal of all of this work and assembling this data is then to be able to combine it with large amounts of genetic sequencing data, and you can generate, meaningful interpretations through genomic-wide association studies, or GWAS studies. And these GWAS studies then can really predict what parts of the chromosome are responsible for the upregulation or downregulation of a specific metabolite of interest. And with those genetic sequences, markers become absolutely important to identify whether you have the correct sequence present or not. And I'm going to let Chris take over from here and talk about genetic markers.
Thank you, Caleb, for that great introduction and great talk through chemistry of this plant. And one thing I'd like to start out this talk on is how interconnected this whole system is. We can't really do genetics without chemistry. And understanding the whole metabolome is extremely important when you start getting into creating these genetic markers to help you do this marker-assisted selection and breeding. And so you can see on this kind of triangle on the right that the chemicals are interacting with the DNA. The DNA is making proteins that then interact with the DNA as well. The chemistry can interact with the proteins and vice versa. It's a very interconnected system. And so you're really looking at how all these things work together, and then also what is the thing we want out of this. And so as we saw on that last slide, there are some rare types of cannabis or smaller clades that are less populated than other areas of cannabis. And so once we find something that is rare or something that's desirable, how do we then take that to something that'll end up benefiting a end consumer or a breeder? And so genetic markers are a great way to kind of bridge this gap. That chemometrics can do a lot of this, that we can see basically all the chemistry that's going on in this plant. However, when we get into these crosses where we want to screen a couple hundred or a couple thousand plants to find this really rare phenotype, it's going to be very costly and time consuming to do a single chemistry sample for each plant. And so that's where genetics can help you take a smaller amount in a faster way. You spend less time doing it, and at a cheaper cost will let you screen more plants, and you can have a better shot at finding that rare phenotype that you're really looking for. And so while not all chemotypes are easily reproduced, there are some underrepresented types of cannabis that can be bred for using these genetic markers. And so that's something that we really look at here at Front Range, looking at the correlations and saying what is common here and what is really rare. And then saying, okay, if it's really rare, can we actually produce that, or is that something that there's a reason why it's really rare? And so we get into understanding the associations of the DNA with the proteins and the chemistry. And that's really how you can kind of relate all of this together and then make a marker-assisted breeding selection. And so when we get into genetic markers and how they work, we are really focused on these biosynthetic pathways here. all the secondary metabolites are usually done through these enzymatic biosynthetic pathways. And so each of those genes that are put at the arrows throughout this pathway are things that can be genotyped, or you can tell the sequence that's involved in that gene that then is going to create the protein. And if you have a different gene sequence, you're more than likely going to have a different protein. And so what we're really trying to do is relate the gene sequence to what that protein functionality is. And so being able to say, I know the exact sequence of this gene, you're starting to be able to relate that to how these compounds are made and what you actually see in the metabolome. And so these compounds aren't necessarily new to cannabis. Well, the cannabinoids are pretty specific to cannabis. These similar pathways exist in a lot of other plants. And it's something that people have been looking into these pathways for a long time. So it is very well characterized. However, once you find that phenotype that you really like, it's now the questions of which gene or genes do I target? And once I can target those, then I can start doing the marker assisted selection to reproduce that phenotype in any cross that I do from there on out. And the best way I can kind of put it is just one marker isn't enough. Say we wanted to have a very high CBD cultivar that made over 20% cannabinoids, but very little THC. The CBD to THC ratio would be determined by a different enzyme than the total cannabinoids. And so that's something we always look at, as well as where in this pathway do we need to target to answer the question that we want. And so kind of going over how they work is really targeting these biosynthetic pathways, but you really want to find where to target. And this is something that is an ongoing process always. And it really depends on the question you're trying to answer by creating the genetic marker. So as we said, the CBD THC ratio is done by those cannabinoid synthases at the bottom of that pathway, whereas total cannabinoids may be something more upstream because it really depends on the amount of fatty acids you produce, the amount of adrenyl diphosphate, and some of these upstream precursor molecules. You need a lot of those if you want to make a lot of CBDA. And so I use the example of flavonoids here. This is a newer pathway that just published in the last year or so, that they did a lot of this work in understanding how these compounds that are some specific to cannabis as well, like the cannaflavins, are produced. And so say we wanted to breed a plant with cannaflavin A and camphorol, we'd have to target multiple areas in this pathway to be able to do that, specifically at the breakpoint between dihydrocamphorol and apigenin, where that FNS gene and F3H gene are. You'd have to target both of those because those are competing for the same precursor molecule. And so understanding kind of how the affinity or how much that enzyme likes that precursor is really how we kind of go about figuring out where do we target and what do we really care in these selection processes. And so it kind of relates back to the rate limiting step in this pathway. There's always some point that you can't make a lot of apigenin unless you have a lot of melanoCoA, for example. And so you have to really look up the pathway and see where is there an abundance of this precursor, and then I have a rate limiting step usually after that. And so we really breed for this enzyme efficiency and activity, because some of these enzymes also will alter activity, particularly like terpene synthases. They're very similar to each other, but small nucleotide differences can really change the product profile coming out of them. And so once we find a gene that we like and we know that this is somewhere we want to target, we'll have to go look into that gene more and find something unique that we can go target. So we really try and find a region of a gene that's informative, usually has SNPs in it or single nucleotide polymorphisms, and they differ from strain to strain. So we're able to relate that SNP to a given function or a given chemotype of that, the variety that we're breeding. And it has to be unique within the genome. And that's easier said than done. It's a 3.5 billion base pair genome. So you're looking usually for about a 20 base pair stretch that only happens once in the genome. And so that's kind of the idea of how we make primers, is this small little complementary strand of DNA will bind to one spot in the genome. And then we can put two of those and amplify everything in between. And so when we get into, we find the gene, we find where in the gene we want to target, then we go ahead and we make different kind of endpoint technologies or ways to report this to an end user who's trying to breed. And so PCR primers are kind of the most common type of primers we use. Polymerase chain reaction has been around since the 80s. There's a couple different variants of it that'll help you answer different types of questions that you may have. And so if my main question I want to answer is this gene present? Is the gene to make cannaflavin A present? Just regular PCR where you run an amplification step and you get whether the band was produced or not in the gel, it's a great way of doing it. Or real-time PCR is another way of doing it where you can detect a fluorescence change as the gene copies are being made. And so you can really see in real time how present is this gene? Do you have the right version of this gene? And then the next step on beyond that is you can have the gene in your genome, but it may not be expressed. And so that's where qPCR and reverse transcriptase qPCR come into play. And that's usually done on a transcriptome library. So you'll make an RNA extract and then run PCR on that rather than doing it on a DNA extract. And so that can tell you how much of your gene of interest is expressed. Out of all the copies of genes in the transcriptome, I'm looking for this one and how often is it there? And if there's one plant that overexpresses it and underexpresses it, that's something you can select for now. And so kind of the next step here would be looking at what version of my gene is present. And HRM or high resolution melting is a fantastic technique that we use readily and very commonly in our labs. Shown by the middle picture over there, that is kind of the end result of what you get. DNA has a melting temperature that's based on the number of ATs that are in it versus GCs. And so what you're able to see there is if you have a mutation in one of these genes and you go to try and melt that amplicon or the copy of DNA that you made, you'll see it melts differently depending on the version of the gene that you have. And so you can tell within a simple example of it is a single nucleotide polymorphism where you can have either homozygosity for Cs, homozygosity for Ts, or heterozygosity where you have one copy of the gene with a C in it and one copy with a T. And so the HRM analysis can really help you tease that apart very rapidly. Most of these techniques are done in 96 well formats, so you're doing them in plates and you can get through a lot of samples very quickly at a pretty cheap cost as well. And so that's some of the main advantages of having some of these different technologies. While you're still targeting the same gene or the same allele, you're really looking at what is the question I'm trying to answer here, and then you apply the appropriate technology. Some of the newer techniques out there that are almost a little more relevant to cannabis and in a grow environment where you're having to not necessarily have a giant lab right next to your grow that you can do this type of science are colorimetric testing. And so LAMP, which is a loop mediated isothermal amplification, or TwistDX makes an RPA, which is a recombinase polymerase amplification. And so the bottom picture there is that TwistDX system where it's a little cartridge that you can load DNA into, keep it at a single temperature, and it will either change colors or present two bands or one, and that'll tell you whether or not your gene's present. And so the great thing about this is you don't need a thermocycler, you don't need all the PCR equipment that you would naturally need to do like qPCR or anything like that. You can do it simply in a heat block or a hot water bath and be able to see a color change within 30 minutes of starting the reaction. And so at that point, you can, it takes a little bit more of the bioinformatics background knowledge to be able to design those primers, because you're either looking for a single gene and you can accept multiple versions of that gene, or you get it down to a single allele where you're really looking for this one version of the gene that does this exact thing you want to reproduce this phenotype. A great example would be searching for like a broken CBD asynthase. If you want to knock out CBD production, you can design a LAMP assay that targets that broken gene, and it'll give you a positive color change when you have it or you don't. And so these are the ways that we go about making these genetic tests after we figured out where we're going to test at and what gene are we looking into. And so how does this look in cannabis? Some of the most common ones that we've seen are the ones listed out here. And you can see on the left, there's those tiny little plants, and that's about the stage where we do this genetic selection at. Is once you get those first true leaves past the cotyledon, you're able to take just a half of that leaf and extract DNA from it and predict something about what the full growing flowering plant's going to look like. And so really, we want to save time by not having to grow out these plants, up pot them, do all of this if they're not the exact type of plant we're looking for. And so a good example of that is sexing cannabis. That's been around for probably a better part of a decade now, to where you either want to select only females for production if you're trying to do a flower or cannabinoid production run, Whereas if you're doing a more breeding application, you may want to select those males and isolate those by themselves. Cannabinoid selection has been around as well, similar to the example of lamp that I discussed. There's a couple companies out there selling tests that'll let you select for that CBDA enzyme, or you can select for the THCA enzyme. And if you see that you have both, you'll more than likely be a mixed ratio plant of producing both THC and CBD. Terpene selection is kind of the new and up and coming one that is just being introduced into kind of readily available tests that people can get their hands on and run at home. If you say you really like terpinolene, or terpinolene is really great for a certain condition, that is a certain gene that we can select for there and tell you whether or not this individual will have it. And so say you made a cross of a high terpinolene plant and a not, we can just select the offspring that'll carry that terpinolene gene. And so you don't have to waste time or money searching into those other plants that don't carry the gene of interest. And this can be expanded past just secondary metabolites, or even more customized secondary metabolite profiles that give you some of these extra traits, such as pest resistance. Powdery mildew resistance is one that's coming up, as well as branching habits as well. Those are going to be really important as we get into this regional specificity of cannabis and people wanting to grow it outdoors. We really want to be able to dial in pretty much everything about this plant that we can. And so now we kind of look to some of these other crops to know where we're going in cannabis. We're just hitting this point of rapid growth. And if you look at corn over the last 100 years or so, we see there's this traditional method of breeding where people select really good looking plants, find the best plant in their field, and they'll breed those two together. By doing that, you make small incremental improvements, but you really don't see these giant leaps and bounds of growth, yield, sugar content, things that you care about. You really aren't seeing huge improvements in until we have an understanding that there is genetic diversity out there. And that's something we should prize. That's something we really should hold close to us, because when we inbreed and we like this one selection, makes it very susceptible to being knocked out, such as the case with bananas, where we do a monocropping and we don't have that genetic diversity integrated back in. Once we start introducing that genetic diversity in the 40s to the 60s, we see the slope of this line go up where those red dots are in the middle, and then you start seeing real improvement really quickly. And that's just through improved hybridization of these different genotypes, and you're getting a more robust, resistant, and hardy plant. And then as we start seeing genetic markers and chemistry be used commonly in corn breeding, that slope even goes up farther, and it continually increases every year that you see significant increases in yield, sugar contents, the sweetness of the corn, the resistance that are available to it. And that's something that we have seen up until the last few years. It's still growing. We're still improving this crop and making it more profitable to grow, easier to grow, and just all around a more useful agricultural product that we want in our fields. And so what does this look like in cannabis? As we get to what we're kind of working on now, is building a genomic selection tool. Most of corn breeding nowadays is done through genomic selection. That's something that you, instead of having a marker in a single gene where you just look at, okay, I know the CBDA enzyme is this, you can have markers throughout the entire genome that give you a fingerprint. And that fingerprint will tell you what that CBDAS is, as well as other traits in the pathway too. And so you get a whole broader look into the genome. Instead of breeding for specific traits, we can breed for types of cannabis and more just rapid improvement year after year through doing that. And seed chipping is something that is very common in other crops that hasn't really made it into cannabis yet, where you can take a single seed, chip it, and tell that fingerprint right off the bat. And so we're working towards those things, but the other way of going about this is to continue down this pathway of looking at traits that are desirable and what are things we can breed for right now. As most people know, feminized seed are a really common thing in the industry for hemp and cannabis. And we start seeing hermaphrodites pop up. The sexual dimorphism of cannabis isn't fully evolved yet, to where we do see this intersex populations come up in certain feminized cross. Having a genetic marker that could tell you this is more likely to do that or less likely to do that is a very advantageous thing to have. Other metabolites, we kind of talked about flavonoids and how those are made, but there's a lot more flavonoids than cannaflavins. And so these pathways just keep extending and building on one another. And alkaloids are related right in there as well. And when you think about cannabis in general, it's really those secondary metabolites that are giving you the effect or the medicinal quality that you look for in it. And it can be something as simple as lipid content too. When we talk more about grain types of cannabis, that you look for certain omega fatty acids in the seed. Because if you want to make a granola bar from cannabis, you're really having to breed that secondary or the lipid profile to match the actual application of what you're going for. And kind of like I mentioned earlier, we are seeing regionally adapted cannabis come out, usually through genetics and a combination of genetics and chemistry, really, because we can see pest resistance at, say you have a really bad caterpillar problem, there may be a certain terpene or flavonoid that will repel that caterpillar. And so that's something we've been looking into, of trying to breed these regionally specified crops. Could be as simple as drought tolerance, that you grow in a really low rain fall environment. That's another trait that can be selected. And even out in Colorado, we've seen beet curly top virus through grasshoppers in the Front Range, and that's something certain genotypes of plants will respond better to. So these markers aren't really limited to just a biosynthetic pathway. Some of them can be much more broad and help you just say, okay, every plant I select from here on out will be drought tolerant now. And then I can customize secondary metabolite profiles on top of my already drought tolerant plant. And so that's where these genetic markers can really help you take a step forward in what we're able to select for, how fast can we select for it, and how fast do we get from generation to generation of improvement. And with that being said, I'd just like to thank our whole team here. We have a great team of scientists, as Don and Caleb had mentioned, at Front Range here. And a great team of advisors as well, some from UC Davis that are amazing to work with. I'd also like to thank the Shimadzu scientific team, Bob Clifford and his team. They have been instrumental in getting us the instruments that we need to collect this type of data. And I also added our contact information. If there's any questions that pop up after the Q&A session here, we'd be glad to be in touch and answer any questions you may have. And I thank you for your time today. And do we want to go straight into the Q&A session?
Yeah. So I guess I'm still emceeing, huh? So in the Q&A, we have a couple questions. I don't know if you saw the first question from Zachary Zander. How much do you cross-reference qPCR testing versus environmental factors and stressors?
That is a really good question. And yeah, the way I kind of see it is that your phenotype is a genotype by environment interaction. And so when you can control for the genotype, then you have a better understanding of what the environment really does. So usually when we put a plant out in the field, we're pretty confident of its genotype of, for example, it's CBDA synthase. And so we'll run qPCR on the types of synthase these plants have, and then verify that in 10 or 12 different environments and say, okay, does this actually change now the chemotype we're seeing? Or does this genotype of CBDAS maybe respond better to a wider range of environments? So it's generally something we'll use both pretty commonly. And it helps tease apart what that environmental interaction actually is.
All right, so Philip Zerbe asks, thank you for the presentation. Targeting biosynthetic genes and enzymes is clearly relevant, but can also be limited by constraints in sink tissue capacity and enzyme pathway regulation, such as feedback inhibition in terpenoid metabolism. What are your thoughts on identifying and targeting, for example, regulatory components?
That is a great question, and there's been some recent work into it, and it's something we're also looking into, is understanding the regulatory pathway that underlies this. Because kind of why the multi-omics approach is really important here, because while you can breed for this gene in the genome, like you said, it's not something that's always going to be expressed, or it's not going to be expressed at the tissue that you want it to, or at the level that you want it to. And so being able to look upstream and downstream of these genes and say, okay, outside of the coding region, what is part of this regulatory region? And then you can take a step up from there and look into transcription factors that bind those outer regions of this gene as well as looking into their regulation and even the post-transcriptional regulation, like spliceosomes are a great example of how some of these genes that are expressed can change function really rapidly depending on the variant of how they're spliced. And so it is something that needs to be looked into almost equally as much as the coding sequence. And that is kind of the next step after you can get the coding sequences down, then it's into the regulation and understanding that whole pathway. So fully agree that is the next step here.
All right, thanks, Christopher. The next question is, thank you, from Kelly Maynard. Thank you. Will the papers and studies associated with this also be made available? I think I can speak to that. So the papers and studies associated with this are cited in the talk. But I don't know that we have a mechanism to post them, and there are copyright issues depending on what the copyright status is. So I'm not sure what that means. So they are available and the citations are listed. I'm not sure what more we can do or are planning to do, but I don't know of anything being posted in terms of supporting literature other than citing it.
I would agree with that. Really.
So next question, Erlin Morales. I tried posting a question, may have disappeared. What about the THC misreporting problems that are going on in labs? I'm curious what other shortcuts labs are doing that are causing products to be pushed to shelves that may not be considered 100% safe because of mold, pesticide, and radiation contaminants. Well, so this is really not the right group to be asking that question, although I work in that field. So I'm going to go ahead and address this and say that Front Range Biosciences, these folks, they have their own chemistry instruments. They do their own assays. They're not depending on external laboratories extensively. And when they do require the services of external laboratories, they double check with our own results to make sure that we can trust them. But that whole question is about the testing industry and the regulators that are supposed to regulate the people doing the testing and whether or not that regulation actually takes place. So I don't know that there's anything beyond that these folks can answer, unless there's something either of you want to speak to in that question.
Oh, totally agree.
Yeah. All right, let's see. Looks to me like that is the end of the open questions. Yeah, I don't see any more questions. Anybody else?
I'd say, and if questions pop up after the presentation, feel free to reach out to myself or Caleb. We always love to talk cannabis science, and this is just always a fun conversation for us. We really appreciate the time we've had here today.
Thank you.
Yeah, thank you both for the wonderful presentation. So we will be sending out the announcement for next month's webinar pretty soon. So please be on the lookout for that announcement. I'll see you again in February. I think that's all for today. Thanks, everyone.
Thank you.
Good. Thank you guys.
Thanks, everyone.